All right, here we go. We're doing week 2 of the Genetics 3331 or Biology 3331 Genetics Laboratory. I'm going to go through the PowerPoint slides real quick, and then I'm going to tell you about some links to some other videos that I'm going to post on the YouTube channel so that you can become more familiar with balancers, because balancers are important. Sometimes they're hard to understand. It's good for you to get a good basic understanding of balancers early in the course. So let's start. Of course, my name is Ricky Johnson. You can contact me at rrjohnso, no N at the end there, cougarnet.uh.edu. This should also work if you put central.uh.edu. The problem with me is that I've been at U of H a long time. I have 3 or 4 different accounts. I exist in multiple places at U of H, and sometimes the systems get me confused. It's caused me problems a lot of times with students contacting me. This one should be safe for this semester, so if you contact me at rrjohnso at cougarnet.uh.edu, it should get through to me eventually. It gets forwarded eventually to a correct email that I can read. All right, so plans for this week: more genetics and nomenclature. Nomenclature being like how to name things. I'm going to give you mutant assignments. I'm going to explain that. Setting up first cross. WA1 introduction, which is your writing assignment 1, and of course the introduction to balancers, which can be confusing. I'm going to try and help you out by giving you some more YouTube videos out there that you should be watching so that you can get a feel for exactly what balancers are. So watch for that in an email separate from this video talking about balancers. Alright, so the nomenclature. Sexual reproduction between 2 different individuals, those parents, the P generation, if you want to call it that. They can contain or carry genetic material of each parent. Now, these are the symbols for male and female, so make sure you recognize the left one here is female and the right one is male. Make sure you know those 2 symbols. All right, we're going to talk about allelic inheritance, dominance, and recessiveness. Of course, I covered that a little last week. Gave you some, told you to watch the other videos perhaps on the channel. Determine the mode of chromosomal loci inheritance, autosomal versus sex-linked. Autosomal, these are traits that are not linked to the sex of the individual. If it's sex-linked, it's linked to the it's located on the sex chromosome and therefore sex-linked. How we're going to talk about how the variation in a single species creates and the creation of strains. Strains being those that we manipulate the breeding of the organisms so that you get the traits that you want in them. Think of dogs. You breed dogs until you get the traits you want. You get Dobermans, you get Cocker Spaniels, you get Beagles, all these, Chihuahuas, you get all these different breeds. Well, those are essentially strains in a way. You've bred them until you got the traits you want in the dog, and when you breed them together, they always have those particular traits if you continue to breed them with the same breed. Those are strains. They're all dogs, just like what we have are all flies. We're just breeding them so that they all have, say, white eyes instead of the normal red eyes in flies. So we're going to produce variation this way. All right, so we have the dominant or recessive allele. All right, phenotypes, ratios of offspring will differ. Here we have an example of a dominant and recessive phenotype. Eggs, sperm basically produce these particular ratios. Hopefully you are familiar with Punnett squares, and you know how to read them. You know how to see that you have 100% on the left and 50-50 on the right. So, autosomal. Autosomal are traits that do not have to do with, that are not sex linked. They're in all of the genes that are autosomal, or the chromosomes that are autosomal will be the chromosomes that do not deal with creating the sexual uh, physiology known as male or female. Um, can be kind of confusing in plants, but it's pretty straightforward in animals and insects such as flies or animals. A gene on a sex chromosome is defined as being sex linked, uh, and in humans, of course, it's X and Y. So gene on other chromosomes are defined as autosomal. So the sex chromosomes determine your sex. So the genes on there are called sex-linked genes. The genes that are on the other chromosomes, the ones that don't determine your sex, those are called autosomal. That's all we're saying here. Uh, these are the numbers here for humans, for Homo sapiens. Uh, Understand that it's a little different for flies. In autosomal humans, the X chromosome, for instance, contains about 2,000 genes. In humans, though, the Y chromosome only contains about 78 genes. Uh, this is, we're learning more. Those 78 genes are pretty important. And how these are expressed are actually really interesting, but of course, this course we don't have time to go into all those details. But understand that the X and Y chromosome, their existence in your cells determines your sex. If you have an X and a Y, you are male. If you have two X's, you are female. There are other possibilities, such as when you have three XY chromosomes, three sex chromosomes. But if you have a Y present, typically your physiology is that of a male. If you do not have a Y chromosome, your physiology is that of a female. Right? That's kind of how it works in humans. You have to have two, though, to survive to birth in humans. So you have to have two of them at least. You might have three. All right. There are cases where there were four. But if you have just one of the two, you generally don't survive. The one exception of that is if you have a single X. Those people do survive. Of course, they don't have a Y chromosome, so they are predominantly their physiology is that of a human female, though they do not cannot reproduce. They are sterile. That is called XO. Um, there are um, it's a syndrome, but you may learn about it in genetics. But if you are a female that is born with just a single X chromosome, you have various physiological changes in your body, and you cannot reproduce. All right, so autosomal versus sex-linked inheritance in humans. For a recessive sex-linked trait to be expressed, you do need two copies of the allele. A male needs only one copy. So males only have a single a single X chromosome. The X is what all humans have to have an X. You don't survive to birth without at least one X. If you have two of them, you're female. If you have one, all right, and no other one, you will survive to birth. But if you have one X and one Y, you are male. All right, so an X and a Y are male. That makes you male. And if you have a single X, there is a disorder having to do with that. But like I said, that's a little beyond this class. All right, here is generally how it works. You will have traits that are sex linked. There are videos that I have put out there that help you go through this. But these traits that are sex linked will be on an X or Y chromosome. And here we show how this system works. The sperm fertilizes the egg, and you get the children as you see here. As you can see, there are various ratios. And now notice up here, this is a little darker than these, and then this one is white. So you can see if you are capital N, you have a dominant trait. Little n is a recessive trait. But then there's the one where you have a capital N and you have nothing. You are male and you only get one copy of it. See, the females generally get two copies of everything, and they can be what we call heterozygous. A male can be a capital N, all right, and nothing, or it can be a small n or nothing in this particular, the way they've set this up. If you're a capital N and nothing, you are white because that is the genotype of this. So you can be male, but realize two capital N's or two small n's, that trait can only occur in females. You also can only have one in other words, having two copies of the letter can only occur in females. Males have a single copy of whatever letter it is, and that will often control uh, the phenotype also. Here we're not kind of showing that, but realize that you need to understand that there's a difference between what you look like, your phenotype, and your genotype. And your genotype is what the actual letters that you have, um, the actual genes that you actually have in your cells. So even though both of these here, these blocks, this is a female and this is a male, they both are, notice that the blocks are white. They are capital N here, big N. All right, both of them have that trait, but their genotype, that's their phenotype. So they're white. White is their genotype. I mean, their phenotype. Phenotype's what you look like. But their genotypes, what their genes say, are different. Here, this is two big X's, that's part of your genotype, and two big N's, that's also part of your genotype. All right, this one has an X and a Y, that's a part of your genotype, that's the part of your genotype that makes you male, all right, versus female over here, and then a single N. So that single capital N is different from two capital N's, all right, and then the X and Y portion are another type of your genotype. But your phenotype, they're the same here. They have the same phenotype in the sense of the trait that we're talking about, in the sense of what is your N-ness. If N were the color of your hair, your N-ness here would be the same, maybe white hair, all right? But your phenotype with regard to your sex would be different, all right? So here, your phenotype of being male and your phenotype of being female are different. That's what you look like. As is your phenotype of your genotype. Now, let me make it clear. Your genotype are just what the letters say. Your appearance is your phenotype. And you can have phenotypes that are the same, all right, but have genotypes that are different. And in this particular case, if you ignore sex, these people have, say, white hair, both of them, if that's what big N codes for. All right, and in this case, this person has white hair, but their genotype just has a single N. Whereas over here, you have white hair and you have two N's. All right, so your genotypes are different, your phenotypes are the same. Just trying to make that as clear as possible. Now here you have the chromosomes that a fly has. So we're switching over to the flies, Drosophila, and away from humans. Humans have many more chromosomes. If you remember back here, we have 23 total, and we have an XY sex chromosome, 22 autosomal chromosomes, and one sex chromosome, which can be either X or Y. A little confusing sometimes, but 23 total chromosomes versus 4 total chromosomes. All right, now they have an X and a Y, and then a 2, 3, and a 4. So Drosophila are a little different than humans, one fewer chromosomes. Now, the ratio of X chromosomes to sets of autosomes determines the sex of Drosophila. In other words, it's not quite the same as in humans. It's not just as simple as X and Y or XX and XY. It's the ratio of X chromosomes to the sets of autosomes determines the sex in Drosophila. So, if you look here, a normal diploid male, has two sets of autosomes, X and a Y, and it's male. And you're like, okay, I get that. That doesn't seem weird. We would expect that, right? But it can change. For instance, we go over here and we see this female. This female has 3 X's. Well, we might expect that. We might expect this is a female. I'm skipping some of these more interesting ones. This is a male, but this is also a male, has a single X. So, in other words, normally in a human, the existence of AY anywhere makes you a male. But in flies, realize that it's a ratio. And the ratio of chromosomes is what determines your maleness. And you'll notice that 1X and nothing else, that makes you male. All right. Female, it's a 1 to 1, and here it's like 1 to 2. All right. So 1 to 2 makes you male. So we'll talk about this later, but you need to understand that flies have a different way of determining this. This is a 1, all right. This is a 1, all right, compared to 2. This is a 2 compared to 3, so you get something a little different. Here it's 2. The ratio is actually 1 to 1, and you're female. 1 to 1, you're female. All right, but 1 to 2, in other words, the ratio is sex chromosomes compared to autosomes. Let's go back. 1 to 2 is a male. 1 to 2, see there are 2 of all the other blue ones. 1 orange to 2 of all the autosomes, ignoring this Y here, this Y. So it's the ratio of the X chromosomes. The X is the orange one here. It's not the presence of the Y that actually makes you male. It's the ratio of the X to the number of the autosomes that determines your sex. Understand that. That's very important. Sex-linked recessions are much more common in male flies than in female flies, and males are what we call hemizygous for the sex chromosome. So hemizygous means that your trait is not like XY or XY. It can be, you can only have 1 copy, all right? Homozygous or heterozygous, you have 2 copies of an allele, of a gene, all right? And hemizygous means you have 1 copy of the gene on 1 chromosome and no copies of it on the other chromosome, or you're maybe missing that chromosome, so that's why you only have 1 copy of it. So if you think of it, if you normally have 2 copies of something, like big A and little a, you're heterozygous. But if you have just 1 copy, a big A or a little a, but that's all you've got, the 1 copy, then we call that hemizygous. So if you are male human and you have 1 X chromosome, all right, that you obtained from your mother, and it gives you a trait. Well, that's the only place that trait comes from. You don't get a copy of that one from your father because that's on the Y, and this trait isn't passed on. So you only have 1 copy of A. That would be your hemizygous for whatever A is. All right, there are 3 major mitotic events that contribute to new genotypes of offspring. So you need to learn these. You literally just memorize these. You need to become familiar with the terms of recombination, non-sister chromatids crossing over. Of course, hopefully you know anaphase and metaphase and such things from introduction to bio, and homologous chromosomes we've kind of talked about. And segregation, those things. Maybe the new things that you're not familiar with are like recombination. You may or may not have covered that. Intro to biology you should have, but you may not have. So if I will put out, or I have already put out, some videos regarding this. Now, reciprocal crosses between mutant 1 and 2. Okay, so we have WTM1 and WTM2. All right, so wild type mutant and mutants 1 and 2. So here you're crossing a wild type mutant and a mutant 1 and a wild type mutant 2. So wild types are, well, I guess there could be human wild types. I don't know if I know exactly what one would look like, but originally when we talk about wild type, we're talking about flies. And it does bring up an interesting question. What is a wild type human? Um, but a wild type fly is one that has none of the standard mutations that we study, like they have regular colored eyes, they have regular wings, they have all the regular things that we consider standard in a fly. They don't have any outstanding mutations like curly wings or anything like that. And so wild types are your standard fly. They're a fly without these mutations that we use to study mutation, um, to study genetics. You have different mutants, and they are not going to be wild types. So a fly with white eyes is not a wild type. It has the white eyes mutation. A double balancer, which we will talk about, is a way for you to track these mutations and make sure that the mutations are maintained within your stock. And the reason this is an issue is a lot of times mutants don't breed well, and it's very easy for them to breed so poorly that the the gene disappears out of the pool of genes that you have within your population. In other words, since they don't breed well and the wild types breed better, slowly the mutations are weeded out, so to speak. They disappear from your population. So sometimes you have to track that and to maintain your population so that the mutation exists in there. And you of course have to do that artificially. That would be the naturally occurring thing, is for these mutants to disappear. We also need to discuss complementation test crosses. That'll be later, but we do need to get this concept of a double balancer through this week. So we'll be talking about WT1 and WT2, um, but let's first talk something about Drosophila nomenclature. In other words, how do you name them? How do you refer to them? So a gene symbol is usually a shortened version of a full gene name, and both are italicized. So, for example, D is the gene symbol for the gene dikete, and that means its wings are angled at a slightly different angle than normal. And CY is the abbreviation, if you want to call it that, for gene symbol for curly winged flies. So whenever you discuss these or put these in your paper, and man, you get people lose so many points for not doing this correctly. Um, you need to italicize them and capitalize them. So notice that dikete and D are both capitalized, and CY and curly have a capitalized first letter as well. So realize the letter or letters you use is the shortened version. The first letter is capitalized. If there's only one letter, of course, it's capitalized. And you have to italicize it. So if you don't do that, you can get counted off. Do I catch all of them uh, in your papers? No, but if you do it regularly, I'm catching it for sure, and you'll get points taken off. So keep that in mind. Is that you need to italicize and capitalize the first letter. You need to italicize the whole word or all of the symbols, and you need to capitalize the first one, right? And that is for a gene. All right. Now, other things, proteins and alleles, may be a little different, but this is for a gene, right? For the name of a gene, the name of a mutant. We'll just say for gene. Just leave it at that. Gene symbols. If the gene first isolated with a phenotype with a recessive mutation. Okay, so if it's dominant, all right, it's. I just said one thing and realized there are exceptions to everything here. So realize the recessive phenotype, all right, mutant allele, is lowercase. So that's like your big A and your little a. So understand that the genes, all right, are dikete and curly. They are dominant mutant alleles. So of course they're capitalized like this, all right? And the shortened names would also be, but you have to italicize them, and so these are dominant mutant alleles, which are the ones that we normally deal with. Um, you may not come into contact with something that um, is lowercase, but if you do, um, so this isn't a big issue for us. Um, Allele symbols are usually less than 3 characters. Some of them are 3 characters, but generally it's 1 or 2 letters. Uh, and a plus, whenever you see a plus, designates the wild type allele. So if there is a CY plus, that's telling you that here is a what? A gene with the curly mutation, but a plus means that you have the wild type version of that gene. The CY1 means that you have a dominant mutant allele for that gene. All right, so keep that in mind. Dominant mutant and wild type. All right, now a forward slash separates them. All right, and that means what? Well, it means you're homozygous. Okay, so here, or in this, I'm sorry, it means that these are the two on the slash means these are the two you get. And of course, if you have the same thing on either side of the slash, you are homozygous. Now the first one here is CY plus, CY plus. So your curly plus, well, that's 2 wild types. This is a fly with basically 2 copies of the wild type gene for curly. Now on the second one, you get a heterozygote, which means it has a mutation and it looks to be a dominant one. So it's probably dominant over the wild type. You have to know about them to know exactly how they operate, and they're indicating here. So you have a heterozygote allele for the curly wing gene. You have 2 copies of the gene, of course, in your genome, and one on each chromosome, each of the whatever chromosome it is on, and one of the two is this number 1, and the other one is the wild type, the one that's common in the fly. Now, what we do here, the first type of cross, is that you're going to cross a wild type with a known mutant. And you're going to see what you get. Do you get lots of different types of flies? Do you get all flies looking just like one of the parents? Now, the 2 parents don't look alike. Do their children all look like one parent or the other parent? Do they look like a mix of the 2 parents? That's what we're doing here when we study genetics. You do what we call scores, and you try to figure out the ratios between the various phenotypes that you receive when you breed 2 particular parents. All right, and notice the symbol of virgin female here. All right, you pretty much have to always use virgin females when you cross them in these crosses, and so you have to get them very young. You have to be able to identify them. That's why we talk about it so much. You can't have them interacting with other flies later on because flies can store sperm, so you can't have them interacting with males at all past a certain age. All right, so you have an unknown mutant in a wild type, and here you would find out what that unknown mutant, the nature of it is, and we're going to use something called a chi-square to see if these changes, if you see changes in these ratios, if they're statistically significant. That'll be something we do quite a bit of later on. All right, now the mutant gene unknown is designation UN for purposes of this presentation. So UN is not a real mutation. This is just what we use to kind of designate that we don't know, we're not sure, and it could be capitalized or not capitalized. That indicates dominant and recessive. Now the first genetic cross will be WT1 cross that can discriminate between whether a mutant is dominant or allele. That's kind of what we're working on. In general, the genes that are on the same chromosomes are inherited together, while genes on separate chromosomes are not. I might explain that in more detail to you, why that's the case. You might learn that in genetics, but essentially, if you're on the same chromosome, genes tend to move together, and the closer they are to one another, the more likely they are to stay together. And but that will be later in the course. The unknown here in WT2 is designated with the UN and represented only just for this presentation, as they said. And we're going to try to cross um, a, an, a female unknown, a female mutant, with a male wild type. And so you'll need to produce this cross. All right, now phenotype expression may change. When you become a heterozygote, you may have a different expressivity. Someone like who has one parent has black hair, another parent has blonde hair, and you end up with brown hair. So you kind of express the color. You're not blonde, but you're not. Your hair isn't really dark. It's now kind of in between, kind of brown. So uh, uh, the expressivity and the penetrance needs to be explained. So the expressivity means the degree at which the individual trait is expressed. So if it's not expressed as strongly, like in brown hair versus black, then that is a measure of expressivity. Now, penetrance measures the proportion of individuals that carry the gene. So if you have a group of children, and like I said, with mother has blonde hair and dad has black hair, and there are maybe you have 10 kids and only 2 have blonde hair, well, that would be measure kind of a low level of penetrance because not very many of them had blonde hair. So it's the proportion. So make sure you're aware of the difference between expressivity and penetrance. You have to talk about that in the papers as we move through. Now there is such a thing as incomplete dominance, which I've sort of explained with the brown hair, and you'll need to be aware of that. That should have been something you covered in Intro to Biology, but it may not have been covered. You never know these days. All right, so writing assignment 1, describe the phenotype characteristics of your mutant. So you're going to describe what the mutant looks like. It has white eyes. I mean, that's one sentence, but you need to be a little bit clearer than it has white eyes. Describe the differences between your mutants and the wild types. Maybe even life cycle differences. All kinds of things might go on. The differences between the males and the females. Maybe it's not the same. Maybe the males have even whiter eyes. I don't know what more white is, but maybe they're off-white. The females have off-white eyes, and the males have, really sharp white eyes. That isn't the way it works, by the way, but maybe you could, maybe if you saw something like that, you would need to report that. Um, you need to give your mutant a nickname, all right, and provide a rationale for your nickname. The nickname needs to have something to go with what you saw. So maybe you did see that. Maybe you had a mutation where the females have off-white eyes. The mutant females have off-white eyes, and the males have white eyes. And you don't want to call it white eyes. Maybe you want to call it, what could you call something other than white? You could call it clean and dirty eyes. So maybe you would call this the clean mutation, where the males are very clean, the females aren't quite as, because their eyes aren't as clean, and then of course the mutants have a whole different color, which are the wild type. But maybe you could come up with some name kind of like that, and use it. But that would, you come up. You just have to come up with a rationale. It doesn't have to make complete sense. You just have to have it linked to the name the nickname that you give. Have it somewhat linked to, so that people can kind of remember it. All right, identify 5 possible genes that, when mutated, would give rise to your phenotype. So this is indicating that, hey, there are multiple mutations that might lead to this phenotype or something very close to it. So the degree of curliness of the flies. Or maybe, like I said, maybe there's a mutation for really sharp white eyes, and then there's kind of a uh, beige color that isn't quite white. And those are two different mutations, and it's hard to tell between the two. And you might see multiple mutations that would give rise to at least something similar to what you're observing. So you need to find things that are similar, mutations that are similar to what you're observing, and give a rationale, of course, of why you think you picked the specific mutation you did out of several candidate mutations. And of course, provide a good figure and title legend with your photo. You're going to need to figure out you're going to pick them out of a thing. We of course aren't taking pictures of flies this semester. All right, formatting. One by one to 1.5 texts max. So WA1 is short. All it is to get you used to making pictures and some descriptions and naming things right, because these are the things that people miss, the easy things that people miss on the tests. So, if you're going to miss it here, so you know next time when you get to WA2, or especially you get to WA5, writing assignment 5, that you've already figured this out and don't do it incorrectly. So you're going to learn to capitalize the right things, italicize the right things, put pictures in your papers the right size, use the right fonts, etc. That's what you're trying to do here. And of course, we're going to start with references as well. So don't forget that you have to do references and a legend. Total length, no more than 3, probably 1.5. Actually, they have some things here. This is contradictory. No more than 3 pages. Oh, text. All right, I got it. 1.5 pages of text with a picture, which brings it to maybe 3. All right, so now we're going to talk about balancers. All right, balancers. I will give you additional links to videos that help you with balancers. They are chromosomes that maintain heterozygosity. Balancers exist for all fly chromosomes except Y. All right, and it's interesting why explaining why it is why Y doesn't explains how they work actually, and maybe I'll make my own video explaining that. Balancers contain at least one dominant allele and several recessive mutant alleles. All right, so a balancer is like a region on a chromosome, or some would think of it as the chromosome itself, that has a dominant allele in it and several recessive alleles like around that dominant allele. How there's a nomenclature for naming them: F for first chromosome, S for second, T for third, and M multiple inversions. The multiple inversions can be kind of hard to explain, but this is just going back to those chromosomes we saw in flies. They don't have 20 plus, 23 chromosomes like we do. They have F, S, and T primarily to work with. And a number of different balancers for the same chromosome. So TM6, um, basically third chromosome, multiple inversions, 6. It has one allele is on a balancer. So this is E1 TM6. That's indicating that E1 ebony 1 mutation is on this balancer. All right, so that's kind of the way the nomenclature, how you would say that. Second multiple, all right, SM1 dominant allele CY. So how do you say this? Well, there are several here. Notice here we have our first maybe non-dominant alleles with small letters. We have Cinnabar 2, for instance, or Spec 2. The second allele, AL, is Aristoteles 1 here. And it's kind of, we're going to look at these numbers and notice how some of these are, well, upside down and reversed. That's going to be a way that we kind of explain ourselves where things are. Um, but I'll give you. We'll talk about this later. Um, this is a wild type. Notice this. This one has what is that? Curly. I forget what PM is. Dikeet. Hmm, I don't remember that one. So there's a lot of mutations in this fly. This is like got several mutations. Uh, so realize that's wild type, and that the second one is definitely not wild type. So here's stubble, SB, stubble. It means the hairs are not quite as prevalent and maybe not quite as long. So wild type, kind of maybe you think of the wild type as they're hairy, but no, this is just wild type. It means this is the normal look, and then stubble just means they don't have as many long hairs, bristles. Sorry, shouldn't call them hairs. They are actually called thoracic bristles. All right, and we need to know the place. So the scutum, the scutellum. All right, the back end of the fly. All right, now here's wild type and stubble near the head. So there are head bristles involved as well. And I think this is where the tame stubble comes from because it looks like um, they didn't grow as long or you shaved this one for whatever reason. So stubble. And um, so this is, we're talking about what these mutants look like. But in this particular case, they are, for this particular cross, are considered double balancers. Um, PM. Now notice the difference in the eyes, shape, color, several other things about this one. So notice that it is recessive, and we're done.